Welcome to monobiology. In this lesson, we're going to learn all about the different types of cell transport. Before we do that, we're going to review a little bit about the structure and properties of the cell membrane. The definition of the cell membrane, what its function is in essence, is to allow materials into and out of the cell. And it is a selectively permeable membrane. Now, it's selectively permeable because of its structure. What I'd like you to do is I'd like you to on this piece of paper you have this document. I'd like you to label the different parts of the cell membrane as per our previous video on the cell membrane. I'd like you to label all the parts. I also would like you to try and write down the types of particles that can and can't get through the cell membrane. When you finish that, move on to the next. Passive transport is the first type of transport we're going to look at. There's two major groups. There's passive transport and active transport, and we're first going to look at passive. And there are two characteristics that make up passive transport. Number one, it does not require any cell energy. It happens without cell energy. Number two, molecules move from an area of high concentration to an area of low concentration. The first type of passive transport, so number one, is diffusion. Now, we already know the definition of diffusion, or you probably do from previous courses. Diffusion is the movement of materials from an area of high concentration to low concentration. So that's the definition that would go here. In these pictures, treat them like this, A, B, and C. It's a passage of time. So these particles are in a trapped container at the bottom of a beaker, and they're let go, and they spread randomly throughout the beaker until they are evenly mixed with the other particles, or, if you prefer, until an equilibrium is reached. Okay, and that's diffusion. Another example would be if I had a perfume bottle at the front of the class, took the lid off the perfume bottle, and released the perfume particles. The perfume particles would spread evenly throughout the class. Eventually, it would take a little while. They would move from an area of high concentration to low concentration. And that's diffusion. And diffusion happens in living systems as well. The second type of passive transport, so number two, is osmosis. And osmosis is simply defined as the diffusion of water. More specifically, it's the diffusion of water across a cell membrane, or a selectively permeable membrane. It doesn't have to be a living thing. Now, when discussing osmosis, there are three different situations we tend to use as a comparison. And remember that when you're referring to these terms, isotonic, hypotonic, and hypertonic, you actually have to be comparing two concentrations to be able to use these words. So, isotonic is a situation in which two solutions have equal solute concentration. Hypotonic is a situation in which one solution has a lower solute concentration, and hypertonic is a situation where one solution has a higher solute concentration. Now, think about it. You can't have one of these without the other. In other words, if one solution has a lower concentration, then the other one has the higher. So these kind of go hand in hand. If you have one, you have the other. Whereas this one, they're equal, so you can have isotonic for both. Now, how do these different terms for osmosis, isotonic, hypotonic, and hypertonic, apply to cells? Okay, so an animal cell in an isotonic solution, in other words, the amount of solute is the same on both sides. Okay, there is going to be no net movement of water. Now, why are we talking about water? Well, we know from past experiences that the cell membrane only allows through small, relatively nonpolar particles. So let's just say that there's an equal amount of salt on the inside and the outside of this cell. It's not the salt that's going to have to move. It's the water that's going to have to move. Why? Because the salt can't get past the cell membrane, whereas we know that the water molecules are able to squeeze through the phospholipid bilayer and get it in or out. However, in an isotonic solution, the water isn't really moving very much because there's equal salt concentrations on both sides. There's no high concentration or low concentration, so nothing needs to move. This is the normal state for cells. This is where animal cells want to be. Now for plant cells, it's the same. There's no net movement of water. However, in plant cells, this is not the ideal situation. In fact, plant cells will become flaccid, which means limp, and the plant will wilt if the plant cells are left in an isotonic situation. So if you've ever had celery where the stalks are not very firm anymore and they kind of start to droop, it can happen to carrots too. You'll understand what the term flaccid means. Okay, let's talk about hypotonic. When animal cells are placed in a hypotonic solution, that means that there's a higher solute concentration on the inside of the cell. So there's more, let's just say salt. There's more salt on the inside of the cell than there is on the outside. Now, if there's more salt on the inside, let's just call it that. There's more of that, sodium, on the inside and less on the outside, then we know that the sodium wants to move to get out of there. But because of the cell membrane, it's going to just bounce right back. The cell membrane won't let it through. So what has to happen instead to try and balance out those concentrations is that water has to move into the cell to try and equal out the concentrations. Now, unfortunately, when water starts to move into the cell, this cell is going to expand and expand. And as it eventually, just like a balloon, if you keep blowing it up, it will pop. Okay, so that's an animal cell. Animal cells don't like to be in a hypotonic environment. Plant cells, on the other hand, do like being in a hypotonic environment. They like to have all of that water in there. Why don't they burst, Mr. Monahan, you ask? Well, that's a good question. It's because of their rigid cell walls. And that is the main reason why plants have cell walls, so they can fill up with water and not worry about bursting. So the plant cell has a cell wall that protects it, and this a plant cell in a hypotonic environment is the normal state of a plant cell. Okay? The last one is a hypertonic environment. Now, in a hypotonic environment, there's more salt on the outside. And yes, the salt wants to move in, but as we know, the salt can't because of the cell membrane. Therefore, what's going to happen is that H2O that's within the cell is going to move out. So there's a net movement of water out of the cell. And of course, if we take all the water out of the cell, it's going to shrivel up. It's going to shrink. If enough water is lost from the cell will die. Now, the plant cell happens exactly the same way, but instead of the entire cell shrinking like you saw with the animal cell, you can see the cell wall stays in place. The cytoplasm and everything else connecting shrinks for the same reason. Water's moving out. In this case, it can cause plasmolysis and cell death. So hypertonic solutions are not good for either of these cells. Hypotonic solution is not good for an animal cell. Plant cells love it. Isotonic is okay for a plant cell, but doesn't love it, and it's great for an animal cell. On this page, what I'd like you to do is I'd like you to try and make some guesses as to how these organisms survive in their environments. What I want you to do is I want you to label C and E. C equals and E equals. Now, it's either going to be hyper or hypotonic. So a paramecium, they live in ponds. Is the cell hyper or hypotonic to its environment? Same with the freshwater bony fish and the marine bony fish. Once you've determined what type of environment they live in, I want you to make a guess as to how they survive in those environments, keeping in mind that cells are either going to lose water or take in water depending on the environment that they live in. How will these organisms overcome that particular problem? Cells shriveling and cells expanding can cause cell death and organism death. Make sure you make your notes on this and bring it to class. We'll go over it. Do it in pencil. Facilitated diffusion is the diffusion of particles across the membrane with the help of transport proteins. So particles that are too big or too charged to get through the cell membrane all by themselves, but still are moving from an area of high concentration to low concentration, like these guys here, high concentration, low concentration, can use the channel proteins or transport proteins to allow them in. Now, transport proteins, channel proteins are one type of transport protein. There are other types too. Channel proteins are the main ones we're going to talk about in Bio 11. We'll talk about all of them in AP Bio. Now that we've looked at all the different types of passive transport, it's time to look at the types of active transport. Okay, so on your sheet, I want you to cross off types of endocytosis, and I want you to put above that active transport. So these three things are actually types of active transport. Now the definition, the characteristics of passive transport were that they didn't require any cell energy and the movement was from high to low concentration. It's the opposite for active transport. They do require cell energy, and the movement of materials is from low generally to high. Not always, but in a lot of cases it is. And there's three types of active transport for Bio 11 that we're going to look at. Notice I skipped a couple pages in the AP Bio booklet. We'll go over those in class. So just copy these notes down and we'll go over the other ones in class. Phagocytosis is the engulfing of large solid particles into the cell. The process is nonspecific. In other words, it can take in a bunch of things. It doesn't matter what they are. Pinocytosis is the taking in of smaller liquid particles, and it's also nonspecific. And exocytosis is kind of the reverse of those two. It's the release of particles from the cell, and it requires energy. These two processes here, oh, don't mind me, these two processes here are types of endocytosis. Endo meaning going into the cell, exo meaning exiting the cell. That in a nutshell is the basics of transport, cell transport. Make sure you bring your questions to class in AP Bio. Don't forget we will go over the rest of the booklet as a group in class.